Thank you, everyone, for standing by, and welcome to our webinar entitled uh, Colloidal Phosphorus and Harmful Algal Blooms in Lake Erie. This webinar series, called Freshwater Science, highlights Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Jim Hood from the Ohio State University. Dr. Hood is an associate professor within the College of Arts and Sciences and co-director of the Aquatic Ecology Lab at Ohio State. Dr. Hood's research focuses broadly on understanding how global change influences aquatic organisms and the structure and function of aquatic ecosystems, with an overall goal of gaining an understanding of how global change drivers influence fresh water organisms and ecosystems to improve our understanding and management of these resources. We're delighted to have Dr. Hood here today to talk to us about his phosphorus work. But before we do start, just a few things about the webinar itself. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20-ish, I will conduct a question and answer session. If you'd like to ask a question during the presentation, please feel free to pull up that chat feature located at the bottom of your screen anytime during his talk, and I'll collect and pose your questions out to Dr. Hood at the end of his presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you great webinars. So without any further delay, I would like to introduce Dr. Jim Hood, who will be presenting colloidal phosphorus and harmful algal blooms in Lake Erie. Dr. Hood.
Thank you, Jill, for that great introduction. And thank you, everybody, for taking some time to come and see this talk today. Before I get started, I'd like to thank our funding sources, and particularly the Harmful Algal Bloom Research Initiative, which supported this work, as well as the co-authors on this project. This was a large interdisciplinary project that brought people together with a diverse set of skill sets. It's one of these projects where no one person could do this alone. So I'm just kind of the bobblehead for everybody. So today I'm going to talk about colloidal phosphorus and harmful algal blooms in Lake Erie. And this is going to be kind of a different talk because it turned out to be a very different project. It took a different path. So here's our team at an early meeting. And this project started basically to sort of challenge a key assumption we were making about dissolved phosphorus and and see how important it was. And so scientists often make these simplifying assumptions to deal with complexity and progress in research and understanding. And often these assumptions are evaluated decades ago. And there's some sort of telephone and change in messaging and simplification of messaging as time goes on. And they require reassessment with modern tools. So this is a story of about a bunch of scientists who, for good reason, sort of evaluated the core simplifying assumptions in their field. And we found that these assumptions, this is the headline, right? We found that these assumptions are just fine for management and forecasting of harmful algal blooms in Lake Erie. So the punchline here is colloids don't really matter for these things within the Lake Erie watershed. But let me tell you about how we got there. So today I'm going to talk about a little bit of background on phosphorus and how we measure it. I'm going to talk about why we might be worried about colloidal phosphorus exports to Lake Erie. We'll talk about our results, colloidal phosphorus in rivers, some methodological considerations, the bioavailability of different phosphorus fractions, and then conclusions. So this project really is focused on Western Lake Erie, which we all know has been subjected to harmful algal blooms since the early 2000s. These blooms are dominated by the cyanobacteria microcystis, which produces toxins which can be harmful to humans, pets, and aquatic organisms. The size of these blooms, but not necessarily the toxicity, is strongly related to bioavailable phosphorus loading from the Maumee River between March and July. And so this is a figure from Stumpf's paper in 2016. It shows bloom severity versus bioavailable phosphorus loading between March and July. And so this suggests that, bioavailable phosphorus loads is a key determinant of bloom size and means that we have to understand the controls on bioavailable phosphorus loading. Now, that's kind of complicated because in freshwater ecosystems, phosphorus is associated with a diverse set of molecules. And there are examples of them over here, including phosphate and iron phosphate, which are important in phosphorus cycling within fresh waters. But as aquatic ecologists, we generally don't measure specific molecules like phosphate. Instead, we use operational approaches to divide up phosphorus into different meaningful subcomponents. And so because this becomes so important later, I'm going to walk you slowly through this. This may be very review for some of you, but some of you it might be new. So we start with total phosphorus. That's all the phosphorus in this water sample, including the phosphorus bound to algae, bound to sediment, and in the water. And then the first thing that aquatic ecologists do is fractionate that based on size. So we use a filter that's 450 nanometers or 0.45 microns, and we push the water through that filter. The stuff that comes out is considered dissolved phosphorus. And then the stuff that is captured on the filter is particulate phosphorus. And so just to kind of put this in scope here, this chart goes from 1 nanometer all the way up to 1 millimeter, an ant and a 1 centimeter baseball. And basically we're cutting this off here. So everything in this fraction here is algae and sediment and bacteria. And then this fraction, we call it dissolved, but we also know that it contains viruses, DNA, and some small molecules. The next step is to digest that water into two fractions. And we use a combination of heat, potassium persulfate, and acid to break apart these phosphorus molecules and convert them to phosphorus. These are standard methods that everybody uses, I should say. Within this dissolved fraction, there's multiple ways to break these up, but I'm only going to focus on the dissolved fraction. Within that fraction, we can use analyses, different reactions to analyze different parts of this. The most common analysis is dissolved reactive phosphorus or soluble reactive phosphorus. People often say this is phosphate as a simplifying assumption, but it's really any phosphorus that reacts with molybdate in a weak acid. So that includes small, less than 450 nanometer inorganic and organic particles. The other part of this total dissolved phosphorus fraction is often called dissolved non-reactive phosphorus, or sometimes dissolved organic phosphorus. And so this fraction contains small particles that do not react with molybdate in a weak acid. Note that it's totally operational. We're not measuring anything specific. We're just measuring kind of like what phosphorus reacts with something else. And this often contains more complex and less labile P molecules. And there's no direct way to measure this. Instead, we determine this by the difference between total dissolved phosphorus and DRP. So as you may have noticed, right, this is not completely dissolved. And within this fraction are things called nanoparticles or colloids. I'll use colloids throughout this talk. And colloids are natural, very small particles between 1 nanometer and 450 nanometers. So here, composed of organic and inorganic materials that vary in size from the smallest bacterial cells that we'll find to viruses and DNA and small molecules. Colloidal phosphorus can be found in both of these fractions. So I'll talk about colloidal DRP and also colloidal non-reactive dissolved phosphorus. To separate these fractions, we used these centrifugal filters to basically separate them into something between 1 nanometer and 450, and then what we're calling a truly dissolved fraction, which is less than 1 nanometer, shown here. And so this gives us a number of fractions. We have this truly dissolved fraction, which we presume is mostly phosphate. We have this colloidal DRP fraction, which contains small organic and inorganic particles, again, likely reactive. And then we have this non-reactive fraction that's generally contains particles, that organic and inorganic. So why should we worry about colloidal phosphorus exports to Lake Erie? Let me tell you why we were concerned. And it all really comes down to phosphorus bioavailability and their role in promoting HABs and the role they play in both the management and the forecasting of harmful algal blooms. And this is why I've been talking about these fractions for the last while. So we can start here at the bottom. This particulate phosphorus fraction, at least within the Maumee River watershed, averages about 26% bioavailable phosphorus. The non-reactive dissolved fraction averages or runs between very low bioavailability to moderate bioavailability, but it's highly variable because depending on what kind of particles are in there. Some colloids are bioavailable, others are highly recalcitrant. Colloidal DRP is the same, although we would expect that to be slightly more bioavailable than the non-reactive fraction. And then this truly dissolved fraction we expect to be phosphate, and we would expect 100% bioavailability there. And so this is important because this figures into how people forecast harmful algal blooms. And so I'll use the example from Stumpf to illustrate that. And so Stumpf used this 26% bioavailability from Baker. And then they also noted that about 70% of the particulate phosphorus actually settles out before it ends up in Lake Erie. So they multiplied those together and they got 0.08%, 8% of particulate phosphorus is bioavailable when it makes it to Lake Erie. They assumed that colloidal DRP and non-reactive dissolved phosphorus were both basically zero. They had data on this fraction, but it was pretty negligible. And then they assumed that this dissolved fraction was 100% bio. And so basically this comes up with this equation that Stumpf used to predict harmful algal blooms. And this is sort of fairly similar with some tweaks to what we see in the harmful algal bloom forecast. So the key part of this work is really challenging this assumption that colloidal DRP is zero and or not bioavailable. And this work that came out by River and Richardson in 2019 suggested that colloidal DRP could be a large portion of total DRP in the Maumee River. They suggested about 50% of DRP loading to Lake Erie is colloidal DRP. And they noted that colloidal DRP was frequently associated with colloidal iron, and those iron phosphorus molecules tend to have low bioavailability. Colloidal DRP should increase with flow. So during these storm events that have the largest, highest DRP loads, those should have the highest colloidal DRP. So this suggests that colloidal DRP is something we really need to pay attention to within the Maumee River. We also had pilot data from a number of different rivers across the watershed. So data during storm events. And so we had some data from the Algal and the Maumee that suggested really high levels of colloidal DRP relative to total DRP. And then, but also data from the Tiffin watershed that suggested low levels of DRP there. And so this suggests that there's high spatial and temporal variation in colloidal DRP that may be important. And so this all matters because it suggests that we could be overestimating bioavailable phosphorus loads. And it suggests that there's a lot of spatial temporal variation in colloidal phosphorus, which if we understood, we could do a better job targeting subwatersheds that were contributing more bioavailable phosphorus to Lake Erie. And then we also thought that a better understanding of colloidal DRP could improve our understanding and ability to predict blooms. So based on all of these things, we put together this big interdisciplinary team, resting on the assumption that there was a lot of colloidal DRP out there in the world. And so first we wanted to just sort of characterize it and understand what was driving its dynamics across a wide range of habitats. We were going to understand what was determining its what it was composed of and how it was changing when it moves from ag fields down to the lake. And then we were going to measure its bioavailability. So let me show you some of these results. The monitoring was led by Laura Johnson and Nathan Manning at Heidelberg University. They focused in on the Maumee at Waterville, as well as two smaller tributaries. And what they found was that colloidal DRP is not 50% total DRP. Average percent colloidal DRP was 10%, and these points here with high percent DRP are actually come from samples that have very low concentrations that aren't important to harmful algal bloom management. So after seeing this sort of first year of data, we went back, we nixed our second objective, and we just decided to focus in on determining whether or not colloidal DRP ever reaches this 50% level, particularly during storm events. So we designed a survey where we focused in on 12 streams across the Maumee watershed, and we focused after taking a sample here at in November of 2023, sort of under low flow. We focused on trying to chase storm events and sample these rivers when flow was the highest. And so this shows the discharge from the Maumee River, and the lines show when our sampling occurred. We sampled 6 storm events. We didn't always hit them. And we got in total 108 colloidal DRP samples that were measured in triplicate. This is really hard work. So Ashlyn Bodecker, the postdoc working on this, was chasing these storms starting in January all through into the summer. And analyzing these colloidal DRP samples is really finicky, as you'll see later. So Ashlyn was working late into the night on these things. So this shows Ashlyn and her friend Isabel filtering at 1 in the morning to make sure we got the best colloid samples we could. So thanks to Ashlyn's work here, we got a wide variety of 108 samples. And this shows our samples for all these creeks versus percentile flow when she sampled. And so what we found is that basically, some portion of these were really during high flow events relative to the 5-year maximum, and others were during sort of lower, more moderate flow events. And so our data is shown here. This is percent colloidal phosphorus. We have high during the high when just high flow was high and when flow was low and moderate. And what you see here is basically, first of all, none of these are at this 50% mark, right? Even during these storm events, we're not seeing colloidal phosphorus DRP hitting 50% or even barely 25%. It's much, much lower. So that's one of the take homes, right? During these storm events, we're not seeing high colloidal DRP concentrations. So that we wanted to be sure of that, though. So we were a little bit worried that during these storm events, as water kind of goes up over time, DRP would also go up. But we were worried that colloidal DRP was peaking like maybe before this or after this, and we were missing it. So Ashlyn went out and sampled the Algal River throughout these storm events, collecting samples every couple hours as the storm went through. She did this for 3 storm events. I don't have time to show you this, but basically the results showed that we weren't missing anything. Colloidal DRP was going up with total DRP, and there wasn't a huge problem. So what might be driving this distinction between what we saw in Rivers and Richardson? Well, one hypothesis is that 2023 and 2024 were wet enough. So this just shows percentile flow in the Maumee River at Waterville. 2023 and 2024 had a lower median flow than any other year except for 2002 and 2012. It's also possible that the composition of DRP has changed between these studies due to some process we don't understand, or there could be methodological differences between our approach in River and Richardson that cause some of these problems. And that's kind of what I want to focus on for a second. So they used a variety of approaches. They centrifuged their samples. We tested that. That didn't cause a problem. But they also froze their samples before centrifuging them to remove colloids. And that's very similar to what we did in our pilot data. So we started basically collecting samples that we would freshly filter at night. Ashland was doing this way into the evening, and then other samples that we would freeze and filter later. And so I want to show you those tests. This is colloidal DRP if the sample was frozen versus colloidal DRP if the sample is fresh. And if freezing has no effect, we expect all the samples to fall in the 1-to-1 line. And that's not what we saw at all, right? This is a big mess. I want to draw your attention to two things. First, these samples here in red, these are all samples that were below our detection limit. So essentially, freshly measured colloidal DRP was 0, but when we froze it, we got anywhere between 0 and our highest colloidal phosphorus measurement ever. So if we freeze these things, colloidal DRP comes out of nowhere. Second, when there was colloidal DRP, there are more points above the line than below, suggesting that there tended to be an increase, but the effect of freezing was really complex. Ashland did some modeling that suggested that this was really associated with changes in pH, but also changes in iron concentration in silica. All right, so finally, I want to talk a little bit about bioavailability of these fractions. And this work was led by Justin Chafin at Stone Lab and Alex Kushner and Kelly Peterson, two really fantastic undergrads that Justin was working with. And so to measure bioavailability, Justin and Alex and Kelly would collect water from the Maumee River at Waterville. During 2023, they captured, lower flow events. And then during 2024, they really worked hard to capture higher flow events. They brought that water back to the laboratory. They 450 nanometer filtered it, and then they also 1 nanometer filtered it, basically to remove all of the colloids. And then they spiked it with different levels of phosphorus, and then also with pea-starved algae, and looked at how algae grew based on the level of phosphorus to assess the bioavailability of that. And I don't have time to show you all their results, but I can kind of summarize what they found. First, they found that Maumee River DRP was not 100% bioavailable. Most of the bioavailable dissolved phosphorus was associated with truly dissolved DRP, the fraction less than 1 nanometer. As expected, colloidal DRP did have a lower bioavailability than truly dissolved DRP. And then, somewhat surprisingly, that non-reactive dissolved P fraction was more bioavailable than expected, but still less bioavailable than the truly dissolved fraction. So to kind of conclude, what our results suggest is that managers and HAB forecasters focused on Lake Erie do not really need to worry about colloidal DRP. During this study, colloidal DRP concentrations were much lower than 50% of total DRP as predicted. However, the years we studied were relatively low flow years, and we would expect that during higher flow years, greater precipitation, we could get more colloidal DRP. We also showed that ultrafiltering the frozen samples is not a reliable way to measure colloidal DRP. Justin's work showed that colloidal DRP was bioavailable, but less so than truly dissolved DRP, but the concentrations were low, so it doesn't really matter for the context of management. So this indicates that there's no need to change monitoring or management to account for colloidal DRP. However, given how dry things were during the 2 years of the study, we might want to keep an eye on colloidal DRP in the future. And with that, I'm happy to take any questions. Thank you for your attention.
All right, we've gotten some great questions, so let me start out with a few here. All right, the first question that we had was, what is the DRP level in raw sewage and or treated sewage entering the Maumee River for colloidal or DRP? Sorry.
Jill, we don't measure sewage exports a lot, so I don't really have that number in my
Okay.
fingertips. I'm sorry.
Okay. Do you see the effect of available phosphorus on toxin production as well in addition to the bloom size?
we again, we don't do a lot of this stuff. My understanding of this is that one of the bigger drivers of toxin production is nitrogen concentration and not phosphorus level. But sort
Okay.
of outside of the scope of what I do.
Okay. Um, Sorry about that. Okay. And then another question is why the particulate phosphorus was more bioavailable than the colloidal phosphorus.
Um, I I don't so I guess my answer to that would be I'm not sure it necessarily is. Like the number that I gave for particulate phosphorus was 26%, which is sort of an average from Baker, but that's fairly variable. And these colloidal values, colloidal bioavailable phosphorus, uh, the bioavailability of colloidal phosphorus is also highly variable, right? Because the particles that are actually in this are changing seasonally, and it's really what's in the water that's determining how bioavailable it is.
Okay. Another question we had was, um, what is the relationship between phosphorus and nitrogen in growing your algae?
Um, well, I'm not really sure where that's coming from, but phosphorus and nitrogen are both common limiting nutrients for algae, and so algae need both of them, ideally in balance. And the ratio of those two things can influence how well algae grow and also what algal species you'll get.
Okay. Um, I have one more question. I'm just double checking to make sure if there were any other questions that I missed. Um, oh, can you repeat what DRP stands for? Dissolved.
Dissolved reactive phosphorus. Or if you're not from the Ohio region, you probably know of it as soluble reactive phosphorus. It's
All
it's
right.
it's got about five names.
All right. Okay, so then oh shoot. Okay, there's a couple more. Do
Yeah,
you what type of algae do you use for the growth studies?
I can't remember. Justin led that, and I don't have that at my fingertips. I'm sorry.
No, no, that's okay. Um, all right, I have another question that this might be a question more for Justin than you, Dr. Hood. Are you ready?
Yep.
All right. Um, I The non-reactive dissolved phosphorus was more bioavailable than you had thought. Were the cultures, um, oh shoot, sorry, it just moved on me. Um, were the cultures, did they potentially have bacteria that had exoenzymes? Sorry, like I'm getting all these questions, so then it flips. Um, Exoenzymes that might be degrading things. Do you need me to say that again?
That is a really complicated, detailed question, and I think I would recommend that that person reach out to Justin and I.
Okay.
It's yeah, it's like I don't think it. There are a lot of processes that go on in lakes that make organic phosphorus. So basically non-reactive dissolved phosphorus available to algae. Um, and Justin would be the one best equipped to answer like which of those processes were actually happening in those bioassays.
Okay. Okay. I did get something from Justin, a phosphorus-starved green algae. Um, and it's moving again on me. Sorry about that. Um, this is not a a good feature of Zoom here. Um, I cannot find it now. All right. Sorry. He did he did
You answered it, but
reply.
you can't find it. Yeah.
Yeah, so yes, I would say that person reach out to Justin
Uh,
Chaffin.
Justin said a pea star of green algae that cannot store phosphorus intercellularly. Yeah.
All right. All right. Thank you. All right. And then how important is the watershed land use to the importance of colloidal phosphorus? If this was a mostly wooded watershed or more wetlands, would you expect more or less? We're giving you some really hard ones, aren't we?
Yeah, yeah, this is really hard. So to be honest, we expected a lot of we came into this project expecting a lot of colloids. And as you can kind of see from this talk, um, worked hard to convince ourselves that there weren't a lot. Um, and there's a fair amount of work out there from a wide variety of habitats, including forests, forested watersheds, and streams, um, and streams and ag-dominated watersheds where you see a lot of colloidal phosphorus. And my sense of this is that we don't really have a good understanding of where you're going to see a lot of this and where you're not going to see a lot of it based on land use, or that the patterns in land use are more complicated than that. And I think like one thing I really want to emphasize, because we have a lot of people from outside of like the Lake Erie watershed coming here, is that these results apply to this watershed, but um, there's I wouldn't go to over to a watershed in the Mississippi Valley and just assume that colloidal DRP would not be important there. I think it's something particular about the Maumee watershed that's leading these low concentrations.
All right. Well, that was one of my questions. So you answered that one before I even
Yeah, got me.
asked it. So
Yeah,
that was great. Okay, another question we had. If you have a few more minutes, I have a couple more.
yeah.
All right, perfect. Could the lower DRP numbers during a storm event be lower due to dilution? Would it be better to calculate the loading instead?
Um, I don't. So total DRP goes up during storm events in these river systems, and that's because it's being like stripped off the land, and we would assume that the colloidal DRP would too. So I think if you Ashland's been doing a lot of modeling of that survey data, and it's really there are a couple of the sites where colloidal DRP is lower during high flow than during low and moderate flow. And my recollection is that she's been able to link that to kind of in-stream physicochemical things such as pH more than dilution itself.
All right. One last question. Have you seen a transfer of phosphorus forms between DRP and colloidal forms?
Yeah, yes. And that was originally what we expected the second objective to look into, but we just didn't have enough colloidal DRP to actually see that. But that's what's happening when we freeze these samples. So we freeze these samples and you don't have any colloidal DRP, and then these phosphorus is getting attached to these colloid particles during the freezing and thawing process. So that definitely is something that happens when we freeze them, and also something that I presume happens out in the wild and that other people have seen happen.
All right. Thank you. I think we've gotten all of them. So
Yeah,
let me just
great questions.
you got a lot of really good questions.
Yeah,
Well, thank you. I'll just close here with a big thank you to Dr. Hood for his willingness to talk to us today about his research. This was really an excellent discussion, some really great questions. I did want to remind everyone that the survey URL for this webinar is in the chat feature, so please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue in May with Dr. Steve Lyon from Ohio State talking about his HABRI work on cascading waterways in agriculture. Want to thank you again, Dr. Hood, for your willingness to talk with us today, along with all the participants on this webinar. We hope this was beneficial and hope that you'll join us again in an upcoming webinar. Thanks, everyone, and have a great afternoon. Thank you again, Dr. Hood. This was excellent.
Thank you, Jill. Thank you, everybody, for the great questions and for your attention.
Thank you.